Hi, everyone. Welcome to A Moment of Science, Please. We are a student group with a keen interest in making scientific research more accessible and comprehensible for students through a series of podcasts. My name is Ashley.
And my name is Nancy.
And we'd like to welcome Dr. Jacqueline Beaudry, who is an assistant professor at the University of Toronto's Department of Nutritional Sciences. Dr. Beaudry holds a PhD in kinesiology and health science from York University. Her research interests are primarily rooted in energy homeostasis and fat metabolism. Let's welcome Dr. Beaudry.
Thank you for having me today.
Thank you for joining us. So we'd like to start off with if you could please introduce yourself and tell our audience a little bit more about you and your background as well as your education.
Yeah, sure, no problem. I'm sure like most people in research, the path is always never straightforward. So I'm going to kind of lead you through the path that I've taken to get to where I am today. I started my research journey when I was a second year undergraduate student at McMaster University. And I got involved in helping to clean fish tanks in a behavioral psychology lab. And from here, I went on to complete my fourth year thesis project in biology. And I was under the supervision of Dr. Grant McClelland, where I studied cold adaptations in white and red muscle in the zebrafish. And I really enjoyed my experiments. I continued on in this lab with my master's, where I went on to study the adaptations of cold and hypoxic environments using a CD1 strain male mouse. And we use this as a model. And I was trying to understand how brown adipose tissue in these mice adapt to these harsh environments. And after my master's was completed, I decided to go into the field of more chronic human disease application. And I pursued my PhD at York University under the supervision of Dr. Michael Riddell. And it was here that I studied how high levels of stress hormones, which we call glucocorticoids, or corticosterone, which is the active glucocorticoid in rodents, and the combination of these high levels of stress hormones and high fat feeding in male rats, and how it induces rapid onset diabetes. And I studied how these effects happen at the level of the pancreatic islets, and how it causes major duress to the function of the islets, and how we can partially rescue this phenotype when we use glucocorticoid receptor antagonists and voluntary exercise. And we also studied the effects of high levels of glucocorticoids and high fat feeding on the liver and muscle and on the fat tissue. And so we are currently carrying out some of this research now in my laboratory, where we particularly focus on high levels of glucocorticoids and their role in fat tissue function. And after my PhD, I got my postdoctoral position at the Lunenfeld Tannenbaum Research Institute at Mount Sinai Hospital. And I worked under the supervision of Dr. Daniel Drucker. And here I studied the role of incretin and pancreatic hormones on fat tissue function. And then I started my position at the University of Toronto in the Department of Nutritional Sciences about 2 years ago, so July of 2020. And so that's an introduction to myself.
Thank you so much. That's really interesting to know how you went from cleaning fish tanks as a second year to researching more about adipose tissue and fat metabolism in mice, especially. So I think that's really interesting to know.
Yeah, thank you for the introduction. And so our second question is, you have extensive academic experience as evidenced by the numerous diabetes-related research articles published in your name. So what experiences did you have that allow you to choose your path in the nutritional science field, particularly in biological systems that influence obesity and diabetes?
Yeah, so during my PhD, we studied the combination of high levels of stress hormones, or glucocorticoids, and high fat feeding. And we saw that this phenotype of just giving the rats of high levels of stress hormones actually made the rats mildly hyperglycemic and also induced higher levels of insulin secretion. However, this phenotype was greatly exacerbated when we also combined it with high fat feeding. So we gave the rats a 60% caloric diet that came mostly from fat. And it was incredible to see how the combination of these two treatments induced overt diabetes in these rodents. And also during my postdoc work, we used both low and high fat diets in mice. And it was interesting to observe the differences in the phenotypes with these mice. So these types of studies from my PhD and my postdoc got me interested in the influence of how diets can affect our metabolism, and especially on how fat tissue adapts and responds to these types of nutritional environments.
That's great. So you have worked with both in vitro and in vivo models. Which do you find most interesting to work with? Does one confer more advantages than the other?
Yeah, it's a great question. And to be honest, I do find both models very interesting to work with, where in vitro models help us to study cell-specific mechanisms in an isolated environment. And we can do this with rodent and human cell lines or models. And this can shed light on what's actually happening in a particular tissue of interest. In vivo models can also help us to study how the change in the organism's environment, being nutritional status or the administration of a pharmacological therapy, and how it can affect a variety of tissues and overall metabolism. We now have the ability to remove or to upregulate hormone receptors in the whole body or in a tissue of interest, and then study how the organism responds metabolically to these changes. So this helps us to understand the receptor's function and how it relates to overall energy homeostasis. So in vitro models, the cell line models can be viewed as advantageous because you can culture them and you can study the cells in a particular controlled environment for long periods of time. But sometimes it is the interaction of other tissues and cells that are missing in culture that provide functional change to the cells in the in vitro models may not give a clear picture of what actually is happening in the in vivo model. Where the in vivo models in rodents can help us to understand the organisms as a whole. So it can help us to provide translational data as to what may actually be happening in humans. However, sometimes this may be like, there may actually be species-specific differences, and this may not be completely translatable to humans. So both in vitro and in vivo models have advantages and disadvantages, and we have to learn what is the right model to use in order to answer our specific scientific questions.
That's great. Thank you for the answer. And at the start of your faculty lecture, which for our listeners can be found on our website, you mentioned that hormones play a role in manipulating energy expenditure and food intake. So could you expand on this a little bit more and explain to us how hormones manipulate the two factors, as well as the importance of hormones in obesity and diabetes?
Yes, of course. So as I mentioned previously in my lab, we study how pancreatic and incretin or gut-derived hormones influence how energy is spent and whether or not we want to eat more or less food. So for example, the hormone glucagon is released from the pancreatic alpha cells. And so glucagon is classically known to be released in the blood when sugar levels are low. So when glucagon is secreted into the circulation, it signals to other tissues in the body, such as the liver, that stimulates the breakdown of glucose. And this increase in glucose in the circulation helps us to normalize those blood glucose levels. So glucagon is known actually to increase energy expenditure, and that's conserved phenotype in both humans and in rodents. And this is an increase in energy expenditure is actually measured by the levels of oxygen that's consumed by the organism. So when glucagon is injected into humans or rodents, the levels of oxygen being taken in increases, and thus this indicates an increase in metabolism. So this becomes important to study because we increase levels of metabolism or energy expended by the body, and then this could potentially provide a mechanism of how these individuals become obese, can expend it, and potentially lose weight. So currently we're now seeing in the clinical trials that glucagon is being combined with other gut-derived hormones as it increases energy expenditure and it helps individuals to decrease weight. Another example of how hormones can influence food intake is gut-derived hormone known as glucagon-like peptide 1 or GLP-1. This hormone is naturally released by the gut cells after you ingest a meal, and it potentiates the release of insulin. So GLP-1 is also known to signal to the brain that tells us to stop eating, or tells us that we feel full. And GLP-1 also slows gastric emptying and makes us feel full longer. So people who are diabetic or obese can be prescribed GLP-1 agonists such as exenatide or liraglutide that can help them to lower blood glucose levels and also lose weight, respectively.
Great. So at your faculty lecture, you mentioned 3 different hormones, namely glucocorticoids, glucagon, which you previously mentioned, and GIP. So of these 3 hormones, which do you think influences obesity the most, especially with regards to adipose tissue metabolism?
Yeah, this is a really hard question to answer, as research suggests that all of these hormones do influence obesity in one way or another. So glucocorticoids affect the adipose tissue as it increases lipolysis, which is the release of fatty acids in the adipose tissue stores. And individuals with high levels of glucocorticoids or stress hormones have large amounts of fat, particularly that accumulate around their central region, so around their critical organs. And this can cause some really detrimental metabolic effects. They also have large amounts of fat depots in organs that should not have large amounts of fat, such as your liver and your muscle. And this can be very detrimental to individuals as it elevates their risk significantly for developing type 2 diabetes. Glucagon, as I previously mentioned, increases energy expenditure, and we thought that it did this through an upregulation of mechanisms in the brown adipose tissue. And just to give a little background, brown adipose tissue is different from white adipose tissue in that it doesn't actually store energy in its depots. It expends the energy as heat. So we just don't know how this glucagon actually works to increase energy expenditure, but it might be through mechanisms that are indirectly related to brown adipose tissue. Where GIP is also a very interesting hormone as it expresses receptors that are seen in whole adipose tissues, both white and brown. And if you remove all the receptors of the GIP in the body, then animals have less fat and are more metabolically healthy. But also, if you inject GIPR agonists, and this upregulates GIPR signaling, this acts on the GIP, on the receptors for GIP, then this shows that there's an increase in insulin sensitivity that is linked with adipose tissue function. So GIP-based therapies are now being tested clinically in obese and just got FDA approval in May of this year for the treatment of type 2 diabetes. GIP seems to be important for increasing the body's response to insulin release that helps us to maintain lower blood sugar levels and fat levels. And that seems to be linked with how adipose tissue functions. But we still don't know or understand exactly how this works. So this is something that my lab is currently trying to understand.
Okay, that's great. And so there is a debate in scientific literature with respect to how the GIP receptor affects body weight. And there has been evidence suggesting that blocking or stimulating the receptor results in a decrease in body weight. So which side of the debate are you leaning towards and why?
Yeah, that's a great question. And you are correct. There is much debate as to whether or not it's better to block or to stimulate the GIP receptor. So currently, as I previously mentioned, stimulating the GIP receptor with a GLP-1/GIP receptor agonist is showing to work quite well for patients that suffer from type 2 diabetes or obesity. So these patients that are on GIP-based therapies are able to lose weight, lower glucose and fat levels, and potentially are at risk of developing other comorbidities. There's also significant evidence that blocking the GIP receptor in rodents and in non-human primates effectively lowers glucose levels and decreases weight loss. However, these are yet to be tested in the clinic and in humans. And there is a possibility that these might become better studied in humans, but so far stimulating the GIP receptor is ahead in the game. So we're currently testing both agonism and antagonism of the GIP receptor to develop a more clear understanding of how these mechanisms work and how they work in a chronic disease state. And it's really hard to make a clear decision as to which is the better model, as we don't really know the exact mechanisms of how they work, but so far upregulating the GIP receptor is winning the game.
That's really interesting. It's really interesting to know that this topic is much debated in scientific literature, and I hope more research is conducted so a clear mechanism of these different pathways is better understood. So our mission as members of a Moment of Science, please, is to make research more comprehensible and accessible to students, as well as introducing programs and opportunities for students who want to get involved in research. So do you personally have any tips or advice for students that are interested in pursuing research?
Oh, yes, of course. Doing research is the best would be my advice. No, scientific research is really, it's not always easy or straightforward, but it can be very rewarding. It can be challenging to keep positive and motivating yourself to move forward. And most times it can be a slow process. However, I think it is important to keep what you're doing, to keep doing what you're doing as long as you find it interesting. Always keep asking yourself questions so that you can continue to learn and to improve. Surround yourself by good people that will support you and have similar interests. This will help to motivate you and to keep you focused. It's very hard to do research on your own, and it's so important to work together with others and to stay true to yourself. I find working as a team is a lot easier to do research than trying to do it all, tackling it all on your own. So I do think that it's been very challenging for students during COVID because people have been separated, and it's very important to try to keep these relationships open, even if things have been done remotely. But what I'm enjoying right now is watching the students work together in the lab again and to build these relationships, because at the end of the day, these are the people that help you keep going and to help you to ask those important questions that will stimulate your research.
Okay, thank you so much for the advice. These are definitely helpful for me personally. And by tradition, we're asking all of our guest speakers about their inspirational scientist. So could you please tell us who is your favorite scientist and why?
I don't really have an actual favorite scientist that I can name right now. I meet many new and exciting scientists quite regularly. And I think that the scientists that do inspire me and motivate me are the ones that get me excited from the work that they're doing in the lab. The ones that are able to think on their feet and they're able to overcome barriers of experimental design. I really do enjoy hearing about how scientists are faced with a problem and how they innovatively worked around it to answer their questions, and how they creatively came up with ideas to try to solve the problems that they were trying to solve. So I've been inspired greatly by my mentors, and they have persevered through challenging projects and keep working towards their goals. So that's the type of person that inspires me.
Okay, that's great. Okay, so with that, Dr. Beaudry, thank you so much for coming on today and sharing your research with our audience. It was a pleasure to talk with you. And so this concludes our podcast. Thank you to everyone for listening. And if you found this podcast engaging, keep an eye out for our next episodes, and don't forget to subscribe to us on YouTube and follow us on social media.